I thought today I would get started with just where to find how you can access the data that you're interested in. And I know some people are interested in bacteria and some people are interested in viruses, and a few people are interested in both. So I figured I will try and do some bacterial use cases and some viral use cases. But I'll start with viral because that's my expertise and what I'm more comfortable with. So there's a few different entry points. If you're just interested in generally browsing for data, you can click on one of these general tabs. It's because we have information on bacteria, a few archaea, and mainly bacteria and viruses though. And a few, we have some information on eukaryotic hosts because we use that for our metagenomic services, and we need that. But the main tabs of interest are bacteria and viruses. So if you click on the viruses, this will take you to our viral landing page where you can see general information on viral families. And on this landing page, they're classified by the Baltimore classification system, which basically groups viruses by their mechanism of replication. So single-stranded positive sense RNA, double-stranded RNA, double-stranded DNA, and so on and so forth. And over here on the right, we have a list of viruses that have been the subject of recent outbreaks and are emerging infectious diseases. And so similarly, if you press on bacteria, this will take you to the bacterial landing page where you have some initial tabs that include bacterial genera that are of the most interest to researchers and are the subject of a lot of research. But this doesn't mean that this is all that we have in the database. You can search for, let's say, something that doesn't exist here. Let's say my favorite bacteria is Morganella. And you can do that just by going to the global search button and clicking on Morganella. And this will search for all types of data that's available here. So I'm going to go ahead and click enter. And this will give me a summary of everything in the site that is listed as Morganella. So at the top, we have the summary box. So showing that there's 25 taxa that have the name Morganella, 346 genomes, several 100,000 proteins. We have some specialty genes annotated. There's information on protein structures, one immune epitope only, pathways, subsystems, and genomic sequences. So, and if you want to scroll down and see more details, you can do that over here. This just shows like the top few results, but we'll give you an idea of the type of data that's in these specific subcategories. And if you just click on it, you can explore that more in a table format over here. But let's say you want a more specific type of data and you only want to see how many genomes there are. You can just click on genomes and click on Morganella and search. And this gives you just the genomes that apply to that. So let's see. Another way to access the data is by going to the organisms tab at the top of the page. And again, this gives me a list view of some of the most important bacterial pathogens. And then on the viral side, we have the viral families that are covered in this database. And down here, we have some of our featured viruses. And if you're interested in all the viruses or all the bacteria in the database, you can just click on these. So I'm going to start with, let's say, a global search or I'm just going to go to influenza under influenza A virus, because that has a ton of data and a ton of different data types that we can kind of walk through and talk about. So on the landing page for the influenza A virus page, we have a few different tabs, and these tabs are for the different types of data that's available. And these may change depending on the infectious disease type that you choose, whether it's bacteria, whether it's a specific virus type. So for example, our segmented viruses have a strain tab that lets you view all of the segments for the viruses. When I go to the bacterial example, we can see that there will be a few other tabs as well that are different from what we see for viruses. So there's a little taxonomy summary here giving information about the taxon ID, how many genomes or segments of the virus there are, how many proteins, how many mature peptides there are, which are basically larger proteins that are then processed and cleaved into smaller proteins, and how many 3D protein structures there are in the database for this particular infectious disease. And then over here in this table, we have a few reference genomes and representative genomes as well. And then just a small graphical overview of what's here. And you can switch this to the table view to kind of see the metadata summary as a list if you like. So if you would like to explore the taxonomy of these viruses, you can do that. And I find this particularly useful when I'm trying to do any kind of comparative biology or comparative genomics for my viruses, because it helps me understand where my virus of interest falls in the family. And it lets me specify the specific subgenus or species that I'm interested in. So let's say you're only interested in H5N1 viruses. You can scroll down to that specific subcategory. You can open and close this little triangle here to see the different types of virus strains that are included under H5N1. And if you notice, when I click on these specific tax IDs, you will get a few different options in this green action bar. When you click on this, you'll get a little bit of information on the record that you've clicked, and you'll also get the option to switch to a different view to basically just select all of the information relevant only to this specific tax ID. So you can click on this, and this will switch it to information that's just relevant to the H5N1 subtype. And you can see what you're looking at by looking at these breadcrumbs up here that show you the different subcategories that you're looking at. So if I then move on to the strains tab, this will show me information on not just a specific segment of influenza virus, because as I'm sure many of you know, influenza has, or at least influenza A has 8 segments. And you can see the information on them in these individual columns over here. So we've got 1, 2, 3, up to 8, and it tells you which strains have what sequences available. So if we look at this record over here that I've just highlighted, this particular strain has all 8 segments sequenced. And you can find these by clicking on the genomes. And this will show you all of the records here. Okay. If I go back, let's see, this is a little bit buggy, but sometimes I can just refresh the page and get that back. All right, so if I go back to the strain view, I can again see the information that I'm interested in. So let's say I want these specific viruses that have all 8 segments. And we're adding, again, I should mention that the site is in beta and that we're still working on it and improving things. So one of the things that our developers have been working hard to do is to add a column that tells you how many segments are available. So that's something that you can search for, and you'll be able to select only the viruses that have all 8 genomic segments available. So once you have, let's say, your viruses of interest selected, you can go ahead and choose to group those. And we have a couple of different ways to group things in the BV-BRC. But the 2 main types that you need to remember are genome groups and feature groups. And the genome groups contain basically nucleic acid sequences, and they contain, you know, intuitively the genomes of the virus or bacteria that you're looking for. And the feature groups can contain all annotated features, but generally we use those for proteins, which would be amino acid sequences. So if I click on group, this is going to give me the option to save these specific strains into a group. And I can either choose an existing group or a new group. And again, this will default to the genome groups folder that every user in the BV-BRC already has, or you can specify your own folder and give it a new name. You can make your own folder. Let's say if you want to go to your home folder, and you can just click on this file icon and say, let's say I give this today's date, March 25, create the folder, and you just go ahead and click on it and say okay, and then give your group name as influenza A and add that. So then you might want to be able to, you might say, okay, great, I've made my genome group, but how do I go about finding it? So that's going to be now in your workspace, and you can go to your home folder, and there it is, March 25, and this is the genome group. And if you look at this icon, all of your genome groups will have this particular label, and you can again go to a list view of the genomes that are in there by clicking on view. And again, you have several tabs that will allow you to look at the data. So if I click on genomes, this is going to give me a list of the individual genome segments that are available in this folder. And it says it's loading, but I guess it's taking its time today. All right, so what else can we look at? So I'm gonna go back a few steps to the original tab, and we're back to my H5N1 subtype. And let's see, we've gone through the strains, but if I want to look at the individual segments, I'm going to click on genomes. Now, see, this has right now about 29,000 segments, but there's a lot of information here. There's the genome name, there's the strain name, there's the GenBank accessions, there's the size, and you can see that there's a lot of variety in the size over here. There's small ones and then there's large ones. And it's kind of all mixed up. They're coming from different hosts, different areas. So I might want to filter the data further. So I can go ahead and click on filters. And this is going to open up for me a series of a list of the different metadata that's associated with each of these records. Because if I click on these records, you can see all the detailed information that we not only take out from the GenBank accession number, but that we also curate ourselves and that we add on to the record as well. And so there's quite a lot here, but you can kind of specify exactly what you want by selecting through these different metadata tabs. So let's say you're only interested in complete genome segments. You can go ahead and click on that. And let's say you're only interested in the fourth genome segment for these viruses. Again, you can go ahead and click on 4. And then we've got the subtype and the H type and the N type for the hemagglutinin and neuraminidase proteins, which are important for influenza classification. So you can again use those. In this case, we only have H5 and N1 because that's the subtype that was selected. And let's say we want to see how many of these viruses have been circulating in the past few years. It turns out that there isn't really anything here in more recent years, and that's probably because everyone's been masking and it hasn't really been circulating. But it looks like 2015 was a good year for H5N1, so we can click on that to view all of the H5N1s that were around in 2015. And then if we're only interested in a specific country or a specific geographic group, we can go ahead and click on that. And it looks like most of the viruses that are at least in the database are coming from Africa. So I'm going to click on that. And the majority of these are obviously influenza from chickens in looks like Nigeria. So these are not the only metadata options that you have over here. If you go ahead and click on this gear button, you can customize this filter tab to add a few other metadata fields that you might be interested in. So for a lot of people, the passage information is important for influenza, or you can click on a specific season because the year might not necessarily be important for influenza researchers, but rather the season. So obviously 2015, you could have the 2014-2015 season, or you can have the 2015-2016 season. Let's see what else. You can, there's also information relating to taxonomy and information relating to plate classification. So you can further filter that. And if I close this, I can scroll down and view the different information that's relevant to me. And I can select all of these or none of them. And if you click on, and this is something that I learned that's pretty cool, you can just use the shift button and select as many as you like. So you don't have to individually click each box. Or you can just use this box at the top to select everything. And now you've got, now again, you can group these into a genome group that you can save for further analysis. So you can either add it to an existing group or a new group, which would be, and again, I'm going to specify my March 25 folder that I created and call this H5N1 Nigeria. And you can add that. And then you'll see this little black box come up over here that says that your group has been created. So, so these, now I have a group saved of all the genomic sequences relating to this metadata. What if I want to get the amino acid sequences for this so that I can make a, let's say, a multiple sequence alignment of all of these segments and look at the differences in proteins or the diversity in proteins. So what I would do is go ahead and click apply, because that's really important if I want all of these metadata fields that I've selected to be applied to the protein tab. So I'm going to click on apply. And now when I switch to the protein tab, it's going to show me all of the proteins that correspond to the previously selected genomes based on the metadata that I used. And so sometimes it just takes a couple seconds to load, but if we're patient, here we go. And now we have the individual segments that are available for the hemagglutinin of these H5N1 viruses. And again, the columns have changed a little bit over here. And if you have specific information that you want to see or don't want to see, you can customize that by clicking on this little plus button. And you can look at all of the different metadata options that are available. So again, we're still customizing this. So some of them might not apply to every single virus or bacterial species that you see, but we're working hard to make sure that most of them fit with the specific infectious disease that we're looking at. So for example, we could add the tax ID or the location where this was isolated from, and so on and so forth. So again, if I want to save this as a working set, I'm going to click on it. And in the BV-BRC, these groups are called feature groups. And so once all of these are selected, again, we'll see that the options in this green bar change. And there's a few different things that you can do now. So to create a feature group, you will click on this F button. No, I'm sorry, you'll click on this group button over here. And this will automatically select the group type for you as the feature type. But you can also save the genome group from here as well. And then you can add it to a new group. And again, it defaults to a folder that already exists for every user called feature groups. But since we went ahead and made our March 25 folder, I'm going to do that. And I'm going to say that this is HA for H5N1 viruses and save that. And so once I have that saved, I can go ahead and apply that to multiple different tools and services that are in the database. I can make a multiple sequence alignment from it. I can perform a SNP analysis to look at the different variation, either in the amino acids, which would be my feature groups, or in the nucleic acid sequences, which would be my genome groups. I can use the gene tree tool to make phylogenetic trees, or I can use a couple of other, you can also use a couple of other basic genomic services as well, which we'll cover in our future sessions. All right, so I'll just mention that a few of these other tabs, you can directly make a multiple sequence alignment with something if you just want to view something quickly. But I would not recommend it for large data sets. But if you just want to get a basic idea of what your alignment looks like, you can go ahead and click on the MSA button, choose whether you want the alignment of the nucleotide or the amino acid sequence. And this will open up a new window for you that generally loads pretty quickly and will give you an idea of what your proteins look like. And in this case, they're pretty much identical with the exception of a few. So I'm going to go ahead and close that. Let's see what else. If you just want to look at the FASTA sequences, you can click on the FASTA button over here, which will give you either your FASTA data in nucleic acid or amino acid format. If you want to switch back to the genomes, you can click on the genome view, and you're already in the feature view, so there's not quite much of a point. All right, so if we go back to our H5N1 subtype page, we can look at a couple of the other different data types that are associated with this H5N1 group. And if I click on the protein structures tab, this is going to give me a list of all of the different protein structures that we've imported from the Protein Data Bank, PDB. And again, you have different columns that are relevant to this data search type. You've got the PDB ID, which is the accession number given to the particular structure. You've got the title of the project. So for this one, it would be the crystal structure of H5 hemagglutinin mutant, et cetera, it's coming from influenza A virus, obviously, because that's what we've selected. We have our own internal database ID. We've got the associated UniProt accession number. We've got the gene information. So the majority of these are for HA, but there's a couple of other ones. And all of these columns are also sortable. So you can just click on the top of the column to sort in ascending or descending order and look at the different protein products. So PB2 would be the polymerase basic protein 2, and P is the nucleoprotein, and so on and so forth. There's a couple of different methods that are used to generate structures, and those are viewable here. Previously, the majority of them were either X-ray diffraction or NMR, but more recently, there's been a lot more electron microscopy generated structures, and there's the release date. And again, you can customize these columns by clicking on the plus button and adding the columns that you're interested in seeing or hiding over here. And you can click on the filter button to filter information that is relevant to you. So for example, if you only want to see structures relating to the HA gene, you can go ahead and click on that. And that changes the other tabs that are available here. If you changed your mind and you want to get rid of it, you can just click on this little X over here, and that gets rid of that. All right. So moving back to other information, we're going to switch to the Domains and Motifs tab. And we can see that, again, the titles of the columns change. And so what the domains and motifs are, this is information that's imported from the immune epitope database, IDB. And basically, these are the different immune epitopes that have been either computed, either predicted or computed experiment, or have been verified experimentally. And again, all of these have different sets of metadata that you can kind of read through, take your time. And then you can also filter for the information that you're interested in. So for example, let's say you're only interested in information coming from a specific source, or there's evidence for it in a specific way. You can go ahead and click on that. All right, so actually, I apologize. I think I've confused epitopes and domains and motifs. The domains and motifs are domains that have been predicted by InterProScan that are of particular structural relevance. So for example, specific domains or specific structural motifs that might be of interest. The epitopes tab is the immune epitopes that have been imported from the immune epitope database. And I am not sure why these are not loading. There we go. And again, you can filter these based on whether you want a discontinuous or a linear epitope. You can choose the specific protein. Let's say, again, you're only interested in hemagglutinin immune epitopes. You have a specific host in mind, let's say humans. And then there's also, you can also filter it based on whether the epitope is relevant to B cells, T cells, or MHCs. All right. So there's a few other tabs that are available as well. There's some experimental data, but this hasn't been loaded yet because, again, the site is still in beta, but there's some omics data that will be available under the experiments tab. Influenza in particular has a lot of surveillance and serology data that's associated with it. So again, you can go ahead and kind of explore that by using these different tabs. You can select multiple, let's say, if you're interested in, again, seeing what influenza A has been circulating in, let's unclick this. Let's say we're interested in seeing what's been circulating in the past few years. So I'm gonna pick 2020 because that was the last year where there was a decent amount of influenza surveillance data. And then I'm going to go ahead and specify a specific geographic area, but that's not available here. So again, I can click on the gear symbol and click on, let's see, host or geographic group. And instead of giving me the individual countries, this will give me the geographic, let's see, let's try that again. Right, so that is supposed to give me the specific geographic group. And I think we might have to complain to our developers because this is not working. But anyway, so let's give another example. Let's say I'm interested in anything that was found in ducks. So I'm going to go ahead and click on duck. So anything that was found in ducks in 2020. And so I have here about 12,500 results for the surveillance. And let's say I'm only interested in samples that tested positive, because obviously not every single sample that's taken for purposes of surveillance is going to test positive for influenza. So I select positive, and that gives me just over 1,000 results. So I can select them all. And then you have the option of clicking on this map icon in the action bar to see where these were found on the map. And it looks like the majority of them were isolated from East and Southeast Asia. And what's nice about this map is that you can also add the specific flyways of wild birds, so their migration patterns. So in this case, the ones that would apply are probably, let's see, Central Asia and East Asia. And if you zoom out, you can see that this would be the flyway of migratory birds in Central Asia, and in gray and the dark blue is the flyway of migratory birds in East Asia and Australasia. And this section is where they intersect. But the majority of samples are coming from, or positive samples are coming from either this section or that section. And again, you can zoom in on the specific pin and get some more details there. Or alternatively, you can copy or download this table of records to your local computer. All right, so switching to the Serology tab. This tab will show you information on serology tests that were conducted by specific surveillance groups. And in this case, most of the records are coming from the Sears influenza group, the Centers for Excellence Influenza Research and Surveillance. And again, there's a couple of different filters that you can use to search for the specific data that you want. Again, by host type, host species, common name, collection country. And again, we, especially for influenza, we spend a lot of time on curating the metadata associated with these so that it's uniform, so that it's clean, and to make sure that it's easily interpretable. So let's say if somebody puts in a scientific name, we try to also provide the host common name as an alternative. We provide host groups so that you don't have to look for every single, let's say, duck species name. You can just click on ducks. And that's something that our users really, really appreciate. However, we're not perfect, and there's always a few exceptions. So if you do find a problem in the data, you can please do report it to us. You can click on help and click on contact us and let us know if you find an issue, and we will go ahead and correct that. All right. So any questions in the meantime? Before I go on to specialized searches? All right, I'm not seeing anything. All right, so let's say you don't want to browse through all of the data and trawl through it and go through the filters. You have something very specific in mind. And let's say you're interested in specific proteins relating to, let's say, coronavirus. You can go ahead and go to our searches tab and click on one of these options, depending on what data that you're interested in. So the different options that are available are taxa, which allows you to browse through taxonomy, genomes, so that you can get either viral or bacterial genomes, strains, which again applies to the segmented viruses, including influenza, bunyaviruses, and reoviruses, which includes rotaviruses. The proteins, if you're just specifically looking for proteins, specialty genes, things like antimicrobial resistance or virulence factors, domains and motifs, if you're interested in a specific protein motif or structural domain type, immune epitopes. Again, we've also got protein structures, the pathways and subsystems, which mainly relate to biochemical pathways or specific subsystems in bacteria, and surveillance and serology, which mainly apply to viruses and specifically influenza for the majority of these. So if you click on, let's say, proteins, this will take you to an advanced search page where you can specify the exact data that you're interested in. So let's say SARS CoV, if you want, let's say SARS-CoV-2. You can specify the taxonomy, the taxon name if you know it, or if you can, or it'll predict it for you. So severe, acute, respiratory syndrome, coronavirus. If you have a genome ID, you can input that, although most of you probably won't. If you specifically want, let's say, you want to know about SARS-CoV-2 that has been isolated from dogs, you can do that. And you would do that by selecting non-human mammal. You can specify the specific host name, the specific geographic group if you want. So this would be the major continents for the most part, specific country, let's say specific collection year. In this case, obviously most of them are going to be 2020 to 2022. You can specify the genome length that you want or select whether you only want complete genomes. For SARS-CoV-2, you also have the option of selecting the lineage, which obviously corresponds to the Pango lineage that's been designated. And there's a couple other options that you can use, either the internal BV-BRC ID, although again, most of you won't be familiar with this. And you can also search for the specific gene. In this case, it would be, let's say, the spike protein. And you can go ahead and click on search. And so this probably wasn't a great example because SARS-CoV-2 has such a huge data set that it takes a little bit of time to load. But eventually, what you will get here is a list of proteins that you can select and group together and further process. So it says no results found, but it's still loading. So again, sometimes this takes time. So I'm going to try something that's a little bit smaller for this case. And let's say I'm going to go with an example I gave last week on Crimean Congo fever virus. And let's say I only want GPC proteins, so glycoprotein precursor complexes, and search for those. And so that loads pretty quickly because the number of genomes in the data set for this virus is much, much smaller than what's available for SARS-CoV-2, which is in the millions. So again, you have to adjust your expectations based on what's in the system. So again, you can select all of these and use the group button to save them for further downstream analysis. All right, so let's go back to the home page and let's see, what else have I not gone through yet? So let's say I'm interested in a specific specialty gene, in this case, maybe some kind of virulence factor. So I can say I want to look for anything that's under Escherichia, and I'm interested in enterotoxins. So I can put that over here in the keyword search and go ahead and click on search. And there's let's try that again. All right, so over here, it's given me everything that's available for, let's say, that particular pathogen, but it's a lot to parse through. So I can use this button, the advanced search, to specify something specific based on what's in these columns. So I can choose, for example, to put in here the enterotoxin. Let's see what we get. Oh, did I spell that right? All right, so now we're getting a list of all of the enterotoxins that are available in the database. And again, you can choose to group those together. You can save your group. You can perform a multiple sequence alignment. You can just click on the FASTA button to see the genomic sequences that are associated with these virulence factors. And that just gives you a quick text file that you could just copy and paste if you wanted to. Again, you can group these either as feature group or so let's say new group, and this would save your proteins to enterotoxin and add those. You can click on the pathway button to get a pathway summary of these genes. Let's see, what else? Am I missing anything? you could click on the service button to directly submit these to a service, either BLAST or multiple sequence alignment. And again, these services will vary depending on what information you're looking at. All right. So let me go over my list and check whether I've missed anything. All right, so we've gone over the different types of data, how you can search for data, advanced searches, how to create genome groups, how to create protein groups. Is there anything else, Ron, you think that I should talk about?
I think you've covered most things there. It's pretty thorough.
Okay. Looks like I finished earlier. All right, let's see, there's a question. This is a question about genome groups. In the bacterial genome group view in Patrick, there was a protein family tab that compares all the PGF FAMs from the genomes in the group. That tab doesn't seem to be there.
It will be coming back.
Genome.
It will be coming back. We've had to, because of the amount of genome growth, we're having to refactor the code on the back end and actually turn it into a service rather than deliver it live in real time on the website. So that service is in development right now. The same thing is true for protein families, pathways, and subsystems. But that service is underway right now and should be available soon.
Right. So you might notice that there's some differences. Again, if you go to bacteria, these tabs over here at the top are not necessarily going to be the same as what you see in the viruses. But if there's something that looks wrong, or if there's something that you think we should add, I would really encourage you to click on the Contact Us button and just let us know whether it's a bug, whether it's just something that you want us to add, whether you think there's a way we can improve accessibility to the data. So just send us a note, and we really want to make this site easy to navigate and easy for you guys to find all of the information that you want. So again, let's say, I think the phylogeny tab is not available for viruses. Oh, one of the things I didn't go over is AMR data, which is really relevant to bacterial, people who work on bacterial pathogens. And there's a lot of really useful information here, including the antibiotic types, the phenotype, whether it's defined as resistant or susceptible or whatever in between. And there's some of these that have been verified experimentally, so the evidence would be laboratory methods. And there's some that are predicted by our software, which would be computational methods. There's specific typing methods based on, I guess, which specific antibiotic resistance test you're looking at. And you can kind of specify the specific resistance data set that you're interested in looking at, and you can group the information on these specific strains or genomes that you're interested in, so that you can perform further downstream analysis.
One thing I'd like to, can I add something to that?
Sure.
So a lot of these genomes specifically for AMR, we've read papers and collected data out of SRA and actually assembled and annotated these genomes where they did big studies, but they only deposited, and they did AMR panels, but they only deposited the data in the reads into SRA. And so a lot of these genomes you won't find in this form at NCBI GenBank.
Right.
And we've used this information to build AMR classifiers. We'll have a whole section on that at some point, but just wanted to give you a heads up that this is something a little bit unique.
That's right. Thanks for pointing that out. And then another unique tab to bacteria is the specialty genes. And this one contains information on a couple of different things, including AMR, I think, including virulence factors, and a couple of other things. Is that right, Ron?
That is correct. It's taken a while because
there's a lot.
500,000 genomes. It's all bacteria that we have in the system. But it's things from CARD, VFDB.
There we go.
And they're mapped, in this particular case, they're mapped on by, if it wasn't available already for the genome, it was mapped on through homology with a sort of a high cutoff.
So there's things like antibiotic resistance, drug target genes, virulence factors, specific transporters, and there's different sources. So I think CARD and NDARO for resistance. There's the virulence factor database for the virulence factors.
Stuff we did too that you see in there, Patrick, VF, which are virulence factors we actually curated as part of the Patrick project.
Right, and
last cycle.
Yeah, and if there's a specific antibiotic class that you're looking at, so I know carbapenems have been big lately, so you know, you can just go ahead and click on that. And then I think what else we have? Domains and motifs, epitopes, that's the same. Pathways, these are, this is information that's been curated from, I think, the KEG database. So if you're interested in this
one is going to take a long time. This is the one, one of the ones we're refactoring if you're looking at all the bacteria.
Yeah, I think this would be.
I think the way it's currently constructed now, what it does is if you're looking at a particular bacterial genus, it shows you the, maybe click on, something like that. I don't know if it'll now try pathways. I think what it'll do here is filter down to just the representative and reference genomes there. That's still pretty big. So this is, like I said, currently underway, and then you can look at the
there's over 7 million results just for Campylobacter and 7,000 genomes. So again, if there's a specific biochemical pathway that you're interested in, you can kind of look at the information that's associated with those. And similarly for the subsystems tab.
So those subsystems are ones that have been curated by the Fellowship for Interpretation of Genomes. It's been over the lifetime of the project, and they are they do it from the literature, and it's sort of the back end basis for the RAST annotation system, this subsystem based annotations. And so these are high quality analyses, and when there's an additional analysis that'll be available when we get this tied into the service. So one of the things I want to say about the experiments, the reason you didn't see experiments in the influenza tab there, we do have influenza data in there, experimental data. The problem was right now it's mapped to the host. So all the viral data, experiment data, transcriptomic data is measured through the host. So we're working on a way so that you could find it from the infectious agent as well. That'll be available soon also.